Here we are, AP Biology, Chapter 11, the control of gene expression. We're going to be focused mainly on the role that cloning plays in how genes are expressed or how we create a new organism using cloning. So animal cloning is simply done by doing a nuclear transplantation. And so that means we take the nucleus from one cell and we put it into the cell from a donor, and therefore we use the donor cell just to house the nucleus. But again, the nucleus contains the DNA. That DNA is what's responsible for determining all of the traits. And so we simply follow the diagram here. So what we do is we'll take the nucleus out of one egg cell and we will add that nucleus into a somatic cell from an adult donor. And then what happens is that nucleus now from the donor cell will grow inside of this other cell. And then we form a new individual. So this is the blastocyst. This is very early on in development. And then as we let blastocysts kind of develop, what happens is if we were to implant them into a surrogate mother, that mother would then give birth to the cloned reproductive individual. Or what we can do is take the cells away from the blastocyst stage and we can grow them in culture, which means in a petri dish, and we can control them and use them as stem cells, and we can turn them into specialized cells. We can turn them into cells that play a role in insulin production. We can turn them into skin cells. We can turn them into cells that will ultimately turn into a bladder or a heart. They're using this technology as a way of helping to repair damage from like heart attacks. Now, one of the big things about cloning and why cloning originally started is that it had valuable applications in the pharmaceutical industry. However, it does raise a lot of ethical issues, and that's where it has kind of become a very big topic in biology overall. And here's why. If we take cloning and we use it for non-human mammals, so for example, if we have a pig and we clone it and we make more pigs, people don't seem to mind too much. However, what if we were to take a human and clone it? Does that raise bigger issues? Is there morals? Is there ethics involved in this? What are the legalities of that? However, cloning is currently being used for non-human mammals, so pigs and sheep and goats and things like that. And we're using it for research in pharmaceutical industry, for the medicine industry, and for agricultural in general. Now, there are some huge obstacles that happen and come with human cloning. And so human cloning currently is banned, and so we don't have to worry about that as of right now. However, the therapeutic cloning that happens is the medical benefit of cloning, and this is to produce stem cells, and we're going to spend a lot of time talking about stem cells kind of going forward from here, but here's what happens. Like an embryonic stem cell, so early on in development, once you have a zygote, that has the potential to become every cell that makes up a human being. We have roughly 100 trillion cells that make up a human, and so we recognize that one cell originally divided to make two, those two became four, those four became eight, those eight became 16 and 32. The idea is that somewhere along the line, one of those cells said, I'm going to become a heart, and all of the cells that came from that heart cell became heart cells. There's a cell somewhere along the line that said, I'm going to become bone, and so there becomes more bone cells. A cell says, I'm going to become part of the arm, and all of the things that make up our arm came from that one cell. And so what we're doing is we're finding a way to take our cells and turn them into what we want or what we may need. So some of the current examples that are being used, if we look, here's human embryonic stem cells. So this is coming from an embryo. And what we're doing is we're taking those stem cells and we are cloning them and allowing them to grow up, and then we're differentiating them. We're telling them what kind of cells we want them to become, and we're turning them into nerve cells so that we can maybe repair nerve damage. If someone suffers a spinal cord injury, maybe one day that won't become such a problem because we can give them a new spinal cord and allow their nerve cells to function. Or you can culture them in different conditions to expose them to different traits, and you can turn those exact same cells not into a nerve cell, but now you turn them into a heart cell. And now we have heart cells that can be used to repair damage from, let's say, a heart attack. You can also take these cells again and make them into something different, and you can turn them into blood cells. And so we wouldn't have to have blood transfusions. We wouldn't have to donate blood. We could just make some of our own. Now, the thing about this embryonic stem cell is that there are adult stem cells. If you were to take stem cells out of bone marrow, then what those adult stem cells can do is you can also tell them what to become, and so we could use them to produce our own blood cells. Now, there's a big difference between adult stem cells and human embryonic stem cells. The biggest difference as of right now is that adult stem cells only have the potential to give rise to certain types of cells. An embryonic stem cell could become any cell in the body. In fact, they do become every cell in the body. Whereas the adult stem cells are designed to only replace and repair damage that will normally happen. So we have skin stem cells, and so our adult stem cells can make more skin. Our adult stem cells can be used to repair some liver damage. Our adult stem cells can be used to repair some minor damage that occurs over time. However, it's not unlimited possibilities. Now, one of the things about this is that we are actually hoping by using an embryonic stem cell that we can control the development of it along the way and make it into a type of cell that we want. Well, by looking at that, what we have to realize is that embryonic development is controlled by a series of different gene expressions. So the different types of proteins that are being made changes over time and allows us to make eyes and legs and arms and ears and liver. And so what we're starting to realize is that there's been an early understanding between the relationship of how an organism develops and what genes are being expressed or being made at the time. And the biggest developments have happened by studying the fruit fly called Drosophila. And so what they can do is they can take a normal fruit fly, and you can see that it will normally develop to have antennae develop out of the top of its eyes. Well, if you go ahead and tell these cells, you're not going to make an antenna, you're going to make a leg, what happens is that coming out of these areas, you now have legs sprouting off the top of a head of a fly. And so they realize they can control what genes get expressed, so what develops. And so we're using that to our own advantage right now to make nerve cells, to make heart cells from a stem cell. And so if we were to actually look at the cascade of gene expression that happens that controls the development of an animal over time, what we're going to be looking at is a series of steps. And so here is your overall egg cell, and you can see that there's all these different parts to it. What happens is that gene expression happens that basically tells these follicle cells to start signaling to these egg cells. Now, by doing that, what we're saying is, hey, this is going to become the head region, and this will become the tail region. And so now our cell knows, okay, everything over here is going to look like a head, and everything on this side will look like a tail. And so then what happens is that when you have your embryo, what happens is that the proteins that are expressed in one region of the cell are going to be those that are used for developing parts of the head. And then there'll be proteins that are expressed that then will be used to develop parts of the body. And so what happens is that over time, if we look at the different gene expression, we can make the legs, the antenna, the eyes, the head, the abdomen, the tail, and so on. The genes that are controlling these major parts are called homeotic genes. And homeotic genes do all of the basic anatomy or anatomical parts. And so they are very critical to the development of the proper development of all animals, not just flies, but humans and pigs and sheep and jellyfish and all that. Now, how do they know what genes to make and when to make them? This goes back to cell signaling that we talked about previously from chapter 11 in the Red Book. So chapter 11 here is covering the signaling again as a way of figuring out, well, what is going on? How do we control that? And so here's what happens. There's going to be a signal molecule that comes in. That ligand is going to bind to the receptor. Once the receptor has bound the ligand, then what happens is we're going to have transduction of the signal, and we're going to create these relay proteins. When we create these relay proteins, we're ultimately going to end up expressing a transcription factor. That transcription factor is a protein that will come into the nucleus, bind to the DNA and tell the gene that is that portion of the DNA, hey, we need to be expressing you, so we're going to undergo transcription and translation so that we ultimately create a new protein. That new protein is then part of the expression of creating the leg, the antenna, the heart tissue, the nerve cell. Now, what they have found using research is that these genes that control key development, like this is the head, this is the leg, these homeotic genes are very, very ancient, and they kind of date back through all of the evolution. Now, what happens is that these small genes are actually called homeoboxes, and what they found is that they're very similar regardless of what kind of organism you're in. So if we were to look at a fly chromosome and a mouse chromosome, granted there's huge differences. Within that, there's still the general idea that there's a gene that says this is the head region. There's the general idea that there's a gene that says this is your abdomen, this is your tail. And if we look, you'll see that they have color-coded it here, whereas you can see these orange markings, those indicate the abdomen and tail region. Whereas this green marking, which you can see here, is what ultimately becomes expressed as being part of the head region. And so there are similarities across the way saying this is the head, this is the tail. These are homeotic genes. Now, I believe we're done with cloning.